Hello students, today we will study about secretory tissues. So because in last class we have covered simple tissues and complex tissues. So you know very well about a simple tissue, which is a group of similar cells in their structure as well as they perform the same function. Whereas in complex tissue the cells are of different types in the tissue, but all of them together perform the same function. So next come to secretory tissue or special tissues. So these tissue cells are mainly concerned with the secretion of certain substances or byproducts of metabolism. So secretion means some elimination of material byproducts which are formed during metabolic activities in the cells tissues. So these compounds or products are usually stored in vacuoles, and sometimes they are excreted out from the cells, and they may be stored in certain cavities formed within the cells. So these secretory tissues are mainly concerned with the secretion or excretion of material. So excretion here in the sense, so these byproducts are excreted out of the tissue or cells externally sometimes, such as gums, etc. So in secretions come resins and these oils, etc. So these are secretions and excretions of these secretory tissues. So these byproducts which are secreted or excreted from the secretory tissues in the plants are economically important also. So for example, a rubber is there. So this rubber is formed by using these latex of different plants such as Ficus elastica or Hevia brasiliensis, etc. So the latex, which is the byproduct or secretion of these plants, is used to prepare rubber. And gums, as you know, these gums are exuded from different parts of the body at the point of injury sometimes, which heals at that place in the plant's stems, etc. So in neem and in other species of plants we have seen this exudation of gums at the point of injury, so where the healing takes place sometime, and the infection by different pathogens will also be controlled by the exudation of these gums like that. So gums are much economically important products. So oils, so oils are also secretory products. So eucalyptus oil, there are several essential oils which are secreted by plant cells, and they are stored in cavities within the cells such as eucalyptus oil, etc. So resins, as you know, these are the compounds byproducts which are secreted by the cells in conifers, mainly in pinus. So these resins are stored in the cavities, and these compounds are byproducts which are secreted by secretory tissues, are usually found in different parts of the plant body, stem, sometimes in roots, etc. So there are various types of secretory tissues based on their secretion or occurrence and storage of these secretory products in different parts of the plant. Let us see different types of secretory tissues found in plants. So these are various structures where the secretory tissues are found in plant body. Now let us see first digestive glands. So the name reflects of that digestive glands, the glands whose cells usually secrete certain digestive enzymes. So here, in case of plants, there are some insectivorous plants or carnivorous plants which usually grow in such soils where the nitrogenous compounds are where there is nitrogen deficiency. In such places, they do not find sufficient nitrogenous products or nitrogen. So these plants usually digest proteins of insects. So these plants are known as insectivorous plants or carnivorous plants. So in these carnivorous or insectivorous plants, digestive glands are found in leaves. The leaves in these plants are modified into trap leaves. So these traps usually capture the insects or prey, and inside the trap this captured insect is digested and assimilated by the walls of these traps of leaves. See here, the inner lining of these trap leaves consists of these secretory tissues. These cells of secretory tissue secrete proteolytic enzymes. So these are the digestive glands present in the inner wall of trap leaves, and the cells of this digestive gland secrete proteolytic enzymes. These enzymes digest the body of insects or the proteins present in the insect body. So in this way, the digested proteins are ultimately absorbed or assimilated by the inner wall of trap leaves as such. So in this way, the deficiency of nitrogen in these plants is fulfilled or satisfied by these insects. So here there is one example of Nepenthes and another is Drosera. So in Nepenthes, see here, these are the pictures where leaf lamina is modified into a pitcher, and here the petiole is modified into a tendril, and the leaf tip is modified into a lid-like structure. So this lid attracts the insect or prey, and the in the inner lining of or inner wall of this pitcher there are digestive glands which secrete digestive enzyme proteolytic enzymes. So here when the prey or insect lands on the rim of this pitcher, so when it falls inside the pitcher, again the hair present inside the wall of pitcher, they capture this insect, and due to suffocation or so the insect dies, and immediately the digestive glands get activated and they secrete proteolytic enzymes. So in this way the proteins present in the insect body will be digested, and they will be digested proteins are absorbed by the inner wall of this pitcher. So this is the mechanism of capturing and digesting the proteins of insect in Nepenthes. So here in Nepenthes there are digestive glands in the pitcher. Another example is Drosera. This is also an insectivorous plant. So in Drosera Around this leaf, the leaves are spoon shaped or spatulate, where along the margins of these spatulate leaves there are tentacles or glandular hair. So these glandular hair usually they secrete proteolytic enzymes. So I think you are able to see a drop of liquid which is exuded or secreted from the tip of these tentacles or glandular hair in Drosera. So even in Drosera also, as the insect lands on the leaf, so these tentacles or glandular hair, they capture the insect, and the insect dies or it will be killed. So immediately the digestive glands present in these glandular hair get activated, and they secrete proteolytic enzymes. So these enzymes digest proteins present in that insect body, and at the same time the digested proteins are assimilated by the cells of this leaf. Next, in detail, the internal structure of this glandular hair, you see. So drop of secretion by epidermal layers, and these are the tracheates and endodermis and vascular bundles, where epidermal layers of secretory cells occur. So this is the digestive glands in Nepenthes. So in Nepenthes, these are digestive glands which occur inside the wall of pitcher. So in this fashion they occur. Now, second type of secretory tissue, let us see. Nectar glands are nectaries. So you know nectar, it is just like honey, which is usually sweet to taste. So plants, insects get attracted towards plants for nectar, and they facilitate cross pollination also during their act. So nectar glands are nectaries, possess cells which secrete nectar, a sugary substance, sweet sugary substance. So based on position of these nectar glands in plants, there are two types of nectaries: floral nectaries and extrafloral nectaries. See here in first type, floral nectaries, for example, Rutaceae and Dianthus. See here, the name itself refers to the nectaries which occur in inflorescence or in the flower parts of plant are called as floral nectaries. So nectaries which are usually associated with floral parts or inflorescence are floral nectaries. So nectar glands occur at the base of ovary in case of Rutaceae members. So because it is found at the base of ovary, which is a part of flower, so therefore this is an example for floral nectary. So in case of Dianthus, in Dianthus also nectar glands occur on filaments of stamens. So these are two examples for floral nectaries. So another example under floral nectaries, where we can see in euphorbia. In euphorbia, so these nectaries occur on the margin of involucre in cyathium inflorescence. So in cyathium, the inflorescence is a special type which we call cyathium, where all the bracts of inflorescence are aggregated or united to form an involucre. So there is single female flower and many male flowers of consisting of only stamens, and the nectar gland or nectary is found on the margin of this involucre. So in this way, so this nectary or nectar gland attracts the insects and facilitate cross pollination. So another type, extrafloral nectaries. So in extrafloral nectaries, passiflora. So in passiflora, nectar glands or nectaries occur at the petiole. Osmophores. Next type is osmophores. So cells of osmophores secrete volatile essential oils. So these volatile essential oils also attract insects and they facilitate pollination. So based on shape or form of these osmophores, they may be flap shaped, or they may be cilia, or they may be brush like. So osmophores usually found in orchids. So osmophores also possess secretory tissues. So these secretory cells, so osmophores is a kind of secretory tissue. So the cells of these osmophores secrete volatile essential oils. So with respect to their form or shape, they are of different types. They may be flap shaped and they may be cilia or brushes, etc. So they are mostly found in orchids. Next type of secretory tissue is secretory cavities. So secretory cavities. So these are the cavities or chambers or empty spaces which are formed in the tissues either by death of cells or lysis of cells, or by moving apart of cells by leaving large intercellular spaces. So these glands itself, these two types of cavities are formed in different species of plants. Number one, lysigenous cavity. So this lysigenous, the term itself refers to lysis, means killing of cells or death of cells. Due to death of cells, certain cells, the empty space or or a cavity is formed within the digestive gland. So within this cavity, the secretory products or excretory products are stored. Such cavities are known as lysogenous cavities. And lysogenous cavities can be identified by ruptured walls, cell walls around the cavity. So, around this lysogenous cavity, there are ruptured cell walls because they are formed by the lysis of cells here. So, due to lysis of cells or death of cells, these products, their secretory products are stored, released in the cavity, and they are stored here. For example, citrus and clove oil cavities. See here, in this lysogenous cavity, the cavity is surrounded by the broken cell walls, and due to death and decay of these cells, their secretory products are stored in this cavity, and particularly these oil droplets we can see here in the cavity, which are stored, lysogenous oil glands here given. Next, schizogenous cavity. So, schizogenous cavities are formed by moving apart of some cells, and a cavity or lacuna is formed due to moving apart of cell, and large intercellular spaces are formed. So, these cavities are lined by epithelial lining, or there are no broken cell walls because they are not formed by lysis of cells. Therefore, their cell walls are complete, and the cavities are lined by inner lining of cell wall. For example, in pinus, resin ducts. So, in resin ducts, the ducts or cavities are lined by full layer of cell wall, and in eucalyptus also oil glands, where the cells are moving apart and forming schizogenous cavity. So, this oil is secreted by these inner lining cells, and oil is stored in these schizogenous cavities. Next type of secretory tissue is hydathodes. So, hydathodes are also known as water stomata or water pores. In case of transpiration, the excess water from plants is lost in the form of vapors through stomata of leaves, mostly. To some extent by cuticle and lenticels also the transpiration occurs, but in case of guttation, so another activity in plants where in guttation excess water is lost in the form of liquid drops through the margins of leaves and tip of leaves. So, the loss of water in the form of liquid drops through hydathodes is known as guttation. So, these are the pores or holes, water stomata, which are found at the ends of these veins and at the tip of main mid vein in margins of leaves are known as hydathodes. So, hydathodes are different from stomata of leaves. So, usually stomata are found either on the upper surface or lower surface of lamina, but they are regulated by these guard cells, but the opening and closing of these hydrathodes is not at all regulated by stomata. Here the stomata are present, guard cells are present, but these guard cells do not control the opening and closing of these pores in hydrathodes. Whereas in stomata, the pores are opening and closing is regulated by guard cells. That is the difference between stomata of leaf and hydrathodes of these leaf margins. So droplets of water exude from leaves margins. This activity is known as guttation, the phenomenon we call it. Usually guttation takes place in a humid atmosphere, because in humid weather transpiration rate is reduced, so excess water from the plant body is lost in the form of liquid drops through these hydrathodes. Guttation takes place usually during cool mornings. Example: Colocasia, Lycopersicon, Pothos, etc. So let us see the anatomy of hydrathodes. So hydrathodes, in longitudinal section we can see a guard cell at the pore, but this guard cell is non-functional. It does not expand or contract to open or close the pore of hydathode. So beneath this guard cell there is a substomatal chamber, and next to this there is epithelium tissue consisting of parenchyma cells which are arranged leaving intercellular spaces, and there is a vein ending consisting of tracheids and vessels. So water which is coming out of these vein endings, the water passes through intercellular spaces of this epithelium, and ultimately it reaches this substomatal chamber, and again in the form of drop it will come out of this pore of hydathode. So this is the structure of hydathode which are involved in guttation process. Next, lenticephorous tissue. So lenticephorous tissue is a type of secretory tissue whose cells secrete latex. Latex, which is a type of white milky substance usually. Sometimes it may be colorless also. So it is an emulsion consisting of different kinds of compounds such as proteins, carbohydrates, organic acids, salts, steroids, fats, resins, etc. So here latex is an emulsion where these compounds are not totally dissolved, and they are then separable to some extent. So that is the emulsion. So these solutes are solute like substances, they are not totally dissolved, they are not mixed, so their identity is clear to some extent. That is the emulsion. So lenticephorous tissue consists of parenchyma cells which secrete latex. So latex may be yellow colorless in case of Papaver members, and it is white in Moraceae members. So with respect to storage organs of this latex, usually it is stored in vacuoles, and later on it is released to cytoplasm. So lenticephers are the cells of lenticephorous tissue are of two types. They are latex cells and vessels. Latex cells, these latex cells are usually isolated and they are not arranged end to end, forming a pipe like appearance as such. So these latex cells, they are cenocytic with many nuclei and they secrete latex. In case of latex vessels, where these individual lenticephers or lenticephorous cells are arranged one above the other, and they may be branched or unbranched, so they form a longitudinal row of cells. These are the vessels, and in latex cells they are isolated separately, they are not arranged in the form of rows. So both are cenocytic, consisting of many nuclei, and there are no transfer septa between the nuclei. For latex vessels, example carica, and for latex cells, euphorbia. So lenticephorous cells occur in Apocynaceae members, Asclepiadaceae and Euphorbiaceae, and best examples for latex vessels, Caricaceae members and Compositae members. Next, come to economic importance of latex. So here latex is useful in the synthesis of rubber mainly. So Indian rubber is prepared or it is synthesized by using latex of Ficus elastica. Indian rubber and para rubber. So Indian rubber is prepared by using latex of Ficus elastica, and para rubber is prepared by using latex of Hevea brasiliensis. So papain is an enzyme which is obtained from Carica papaya latex, and opium is obtained from latex of Papaver somniferum, and Asafoetida gums of Acrus sapota, and Asafoetida root stalks of Ferula asafoetida, rubber. So as we have discussed. So today we have covered different types of secretory tissue or special tissue. So once again, so far we have covered digestive glands, next nectaries, and after that we have covered secretory cavities, in that lysogenous cavities and schizogenous cavities, next hydathodes and lenticephorous tissue. So let us stop here. Thank you.